IMBIAN Lab company develops and performs the full production cycle for test systems for human diseases diagnostics. IMBIAN production facilities are located in the Kolesova Science City and is included into the list of the residents of Scientific Production Complex. We present the SARS-CoV-2 IgG RBD IMBIAN ELISA reagent kit, which determines neutralizing anti-RBD antibodies in overinfected and vaccinated individuals. This reagent kit goes in two basic configurations. Every reagent kit includes the instructions for use. Please carefully read the instructions in order to avoid mistakes during the analysis. The kit number 1 includes pre-dilution sample tablet, immunosorbent, sample dilution buffer. Kit includes 2 vials. Positive control sample, negative control sample, conjugate concentrate, homogeneous solution, substrate solution, wash solution concentrate, stop reagent solution, and a sealing paper for microplate. The kit number 1 consists of 2-component conjugate and homogeneous solutions. To prepare the working solutions, the components must be mixed according to the instructions, and the samples must be pre-diluted during the staging. The kit number 2 includes immunosorbent sealed in a foil bag, positive control sample, negative control sample, sample dilution buffer, conjugate solution, TMB solution, a stop reagent solution, wash solution concentrate, and a sealing paper for microplate. The kit number 2 consists of 1 component ready-to-use solutions. Samples do not need to be pre-diluted. The reagent kit is suitable for both automatic and manual assays. Performing the manual test on kit number 2. The reagent kit should be allowed to stand for 30 minutes at room temperature. Add the required number of microplate wells to the plate frame for testing. Place unused microplate wells into the foil bag and store them at +2 to +8 degrees Celsius until the expiration date of the reagent kit. The ELISA procedure begins with the introduction of control samples into the wells. Add 100 microliters of the control to the appropriate wells using the individual pipette tip. Double well control must be used when using 2 or more wells. Add 90 microliters of the buffer solution to each of the remaining wells. Add 10 microliters of extraction sample to the wells. The color of buffer solution must change to blue. Tape the plate. Incubate at 37 degrees for 25 minutes in a thermoshaker. Or, if the laboratory does not have a thermoshaker, the procedure may be done in thermostat for 40 minutes according to the instructions. After the incubation, clean up all wells by washing the wells with the plate washer. Wash the plate 4 times with 300 microliters of wash solution. If necessary, remove the moisture from the wells by tapping the plate on the filter paper. In the next step of the assay, add 100 microliters of the conjugate solution to all wells. Seal it with film and incubate at 37 degrees for 25 minutes in a heat shaker. At the end of the incubation, remove all wells by washing it as in the previous step. Add 100 microliters of TMB solution to all wells. the plate with the foil. Incubate in a thermal shaker for 10 minutes. At this stage, it is important that the incubation is done in a dark place. After 10 minutes, stop the reaction by adding 50 microliters of stop reagent. The color of TMB in stained wells should change. Using the spectrophotometer, we obtain the results in the form of an optical density. Interpretation of obtained data is written in details in KIT's instructions. Calculate the optical density value of the well with negative control sample, which should be not greater than 0.2. The optical density value for the well with a positive control sample should be no less than 0.5. Based on the results, the optical density for the control samples meets the criteria for the validity of the test. Next, to interpret the results, it is necessary to calculate the critical optical density. It is calculated according to the formula: optical density of negative control sample plus 0.2. In this formulation, the optical density critical is 0.236. Samples with optical density higher than this value are considered positive. IgG antibodies to the SARS-CoV-2 RBD antigen are detected. Samples with an optical density below this value are considered negative. IgG antibodies to the SARS-CoV-2 RBD antigen were not detected. The resulting optical density values can be expressed as a coefficient of positivity, which is calculated using the formula ratio of optical density sample to optical density critical. The slide shows coefficients of positivity values for each sample tested. If the coefficient value is equal or bigger than 1, the sample is positive. If it's less than 1, then the test sample is negative. For positive samples, an antibody titer can be estimated using the SARS-CoV-2 IgG RBD IMBIAN ELISA system. For the IgG antibodies titer determination procedure, one control positive sample and one control negative sample each must be used. The upper wells of the stripe, for example A1 and B1, are used for the controls. The control samples are not titrated when staging. Well C1 is filled with 180 microliters buffer solution. Add 100 microliters of the buffer solution to the remaining wells of the microplate. Add 20 microliters of the test samples only into the well C1. Pipette it carefully. Transfer 100 microliters content from the well C1 to well D1. Mix it gently by pipetting 5 times. Repeat the procedure to the last well, inclusive the removing of 100 microliters of the solution after pipetting. Obtain a stripe with 100 microliters dilutions of the test samples in wells. The following steps of the analysis are the same as the procedure for determining the antibodies in the sample tested, which was presented in details earlier. According to the results, the last dilution of the sample that has a value of coefficient of positivity equal or greater than 1 is taken as a titer of antibodies to SARS-CoV-2. In this analysis, the titer corresponds to a dilution of 1 in 40. IMBIAN. We create innovations.